This video is for The Living Earth, Unit 4.1, Genetics. Today's essential question is, how do Mendel's laws help us predict patterns of inheritance? Our first main idea, the law of segregation. Mendel was able to discover a great deal about inheritance from the results of his pea plant studies. He formulated two laws of inheritance. The first, called the law of segregation, deals with what happens to alleles during the formation of gamete cells. Mendel believed that the two alleles for a given character must separate when gametes are produced. Each allele ends up in one gamete, meaning an egg or a sperm cell, and thus each gamete only contains one allele. In this way, when gametes combine during fertilization, an offspring will inherit one allele from each parent. We now know that this is precisely what happens during meiosis. Two partner chromosomes in each pair separate. Each gamete receives one chromosome and thus one allele for each gene. The law of independent assortment. Mendel also wondered whether the expression of certain characters, such as flower color, might affect the expression of other characters, such as pod color. This is similar to asking the question of whether tall people are more likely to have the traits brown or blonde hair. What do you think the answer is? Mendel investigated this question by observing the appearance of two or more characters during some of his breeding experiments. He found that the inheritance of one character did not influence the inheritance of other characters. Today, this law is called the law of independent assortment. This is due to the segregation of alleles during gamete formation. Each pair of alleles segregates independently of other pairs of alleles. Alleles are randomly distributed into gametes, therefore. And this law holds true except when the alleles for two characters are located on the same chromosome. Monohybrid cross. When a genotype for a given trait is known for two breeding individuals, a diagram called a Punnett square can be constructed. A Punnett square is used to predict the ratio of genotypes of the offspring. For example, crossing a homozygous dominant trait, green pod color, for example, the allele which is denoted on the screen here as capital G, with a homozygous recessive trait, yellow pod color, which the allele is denoted as lowercase g on the screen here, we can predict the ratios of genotypes and phenotypes for the F1 and F2 generations. Remember that a homozygous individual has two identical alleles. The results of this cross between a capital G individual and a lowercase g individual are shown on the Punnett square on the screen now. The genotype of one parent appears across the top of the Punnett square. The genotype of the other parent appears down the left side of the Punnett square. The four predicted genotypes of the offspring are highlighted here in magenta. What is the phenotype of these for big G, little g offspring? Remember, because the allele capital G for green pods is dominant to the allele little g for yellow pods, these heterozygous offspring exhibit the dominant phenotype, meaning all of these plants would produce green pods. These heterozygotes are also known as monohybrids. Further crossing them with each other is called a monohybrid cross. Do you remember the phenotypic ratio of Mendel's monohybrid F2 cross? Can you predict it in a Punnett square? Again, all four possible genotypes of the offspring are highlighted in magenta. As you can see, of the four offspring produced by the monohybrid cross, there are two heterozygotes and one homozygous dominant and one homozygous recessive. Just as Mendel noted in his experiments with flower color, there is a 3-to-1 ratio of the dominant to the recessive phenotype. There will be, on average, 3 plants with green pods for every 1 that has yellow pods. Imagine that you had an individual that expressed a dominant phenotype, such as brown eyes, and you wanted to know whether his or her genotype was. Are they brown homozygous, meaning capital B, or brown heterozygous, meaning capital B, lowercase b? Knowing that brown eyes are dominant and blue eyes are recessive, can you determine the genotype of the brown-eyed individual by crossing him or her with another individual, assuming that it is ethical and feasible to do so? A test cross is designed to test this very scenario. In a test cross, an individual with an unknown genotype, in our example, the brown eyes, is crossed with an individual with a homozygous recessive genotype, in this case, lowercase b. The first step is to complete 2 Punnett squares, one in which the unknown individual is homozygous for the dominant trait, and one in which the unknown individual is heterozygous for the dominant trait. In each square, the unknown individual is crossed with a homozygous recessive individual. What would the phenotypic ratios of the offspring be in each case? In the first case, the phenotypic ratio of the offspring would be 100% dominant. In the offspring of the second case, there should be a 1-to-1 ratio of dominant and recessive phenotypes. Using test crosses, scientists can determine the genotype of unknown individuals by modeling the cross of each possible unknown genotype with individuals having a known homozygous recessive genotype. Dihybrid cross. Until now, we have dealt with crosses that involve only one character. How can we diagram a cross observing 2 characters at the same time? Let us take both flower color, remember purple is dominant as a capital P and white is recessive with a lowercase p, as well as pod color. Green is dominant with a capital G and yellow is recessive with a lowercase g. The Punnett square on your screen now crosses an individual that is homozygous dominant for both characters with an individual that is homozygous recessive for both characters. As you can see, all of the offspring for this cross are heterozygous for both traits. They are called dihybrids. So the question is, can we predict the F2 generation outcome of crossing these dihybrids? This dihybrid cross chart must be somewhat more complex to display each possible combination of alleles. So, what do you think the phenotypic ratios of the F2 offspring will be for this dihybrid cross? Looking closely, we find that out of the 16 individuals, 9 are purple flowered and have green pods, meaning both dominant traits. In addition, 3 are purple flowered with yellow pods, meaning one dominant is expressed and the other recessive trait is expressed. Another 3 are white flowered with green pods, meaning the alternative dominant trait is expressed and the other recessive trait is expressed. And finally, 1 individual is white flowered with yellow pods, meaning both recessive traits are expressed. Thus, a 9-to-3-to-3-to-1 ratio is the outcome of a dihybrid cross. Constructing Punnett squares for the crossing of known genotypes allows us to make predictions about the probability of obtaining offspring with particular phenotypes. We can also use simple addition and multiplication to determine this probability.
A monohybrid cross is a cross between 2 heterozygous individuals. Imagine we cross 2 people with brown hair. Both are heterozygous for the hair color gene. We can use a Punnett square to show that the offspring produce both heterozygotes and homozygotes. They'll produce 1 homozygote of one type, 1 homozygote of another, and 2 heterozygotes. What do these look like? Well, in the case of brown-haired heterozygote, they'll produce brown-haired homozygotes, and they'll produce brown-haired heterozygotes. and they'll produce blonde-haired homozygotes. The phenotypes that result from a monohybrid cross segregate in a 3-to-1 ratio. For a dihybrid cross, things get a little more complicated. Now imagine the same cross as a monohybrid cross, but let's consider two traits. Not just hair color, but eye color as well. That's where we need our Punnett square to get bigger, because we have to look at the transmission and segregation of all those alleles for both those traits. In this case, a heterozygote who has brown hair and brown eyes mates with another heterozygote with brown hair and brown eyes. We can follow the transmission through to the next generation. You'll notice that all of the parents had brown hair and brown eyes, but the offspring are variable. Mendel showed that these segregate out in a 9-to-3 to 3-to-1 ratio. 9 individuals have brown eyes and brown hair. 3 individuals have brown hair and blue eyes. 3 individuals have blonde hair and brown eyes. And 1 individual has blonde hair and blue eyes.
Let's take a look at a sample problem. In the dihybrid cross shown on the screen here, what is the probability that the first offspring of the heterozygous parent will be either purple flowered with yellow pods or white flowered with yellow pods? We simply add up the number of individuals that show either of those combinations. Meaning, we have 3 out of 16 showing one of the possibilities, and another 1 out of the 16 showing the other possibility. Simply adding those two fractions together, as we see on the screen here, results in a fraction of 4 over 16, which is the same thing as 1/4, which in terms of a percentage is 25%. What about the probability of not an either or, but an and? So what about the probability that the first 2 offspring born will be purple flowered with yellow pods and a white flowered with yellow pods? In this case, instead of using addition like we just did, we use multiplication. Again, there are 3 individuals showing the first combination and 1 individual showing the second combination. In this case, needing both conditions to be true, we will multiply those 2 fractions, resulting in a fraction of 3 over 256, which comes out to be 1.2%. At this point, you should be able to generate a full and appropriate response to today's essential question. How do Mendel's laws help us predict patterns of inheritance?